your viewers, your listeners, what they need to know about COVID-19 vaccines. It's a difficult story to get right, and our presenters today are going to help you navigate this challenging territory. We will have one keynote speaker to take us through the essentials of vaccine science and safety, and then we will have a health journalism specialist to give you some good ideas on how to approach this topic. To begin with, our keynote speaker will be Professor Bartholomew Akanmori, who is Regional Advisor for Vaccine Research and Regulation in the WHO's Regional Office for Africa in Brazzaville, Congo. He is responsible for vaccine research, regulation, and safety, where he leads a team at the Secretariat of the African Vaccine Regulatory Forum. Dr. Akanmori will guide us through the science and safety of COVID-19 vaccines. If you have questions for Dr. Akanmori, please use the Zoom chat box to type them in. And my co-moderator, Tole Nyata, will scan the questions and he'll pick up as many as we have time for to pose to Dr. Akanmori. Right now, I'd like to hand over to Tole to talk about some community feedback we have.
Thank you very much, Wayne. Christophe, if you share the community feedback slide, please. So these media dialogues have been going on from last year, and they are informed by a community feedback mechanism that is from RCCE, ESAR region. And from this community mechanism, what we get from the ground is what comes up as poll questions, and participants participate in the poll and they help us choose the next conversation. So, Christophe, when you're ready. So we are going to look at the latest community feedback, which is on COVID-19 vaccine development and safety. So this is what we are getting from the ground. With regards to dependency on traditional remedies to treat COVID-19, the feedback we are getting from the ground is we have our own remedies and do not need the vaccines. This was had in 9 countries: Kenya, Tanzania, Ethiopia, Uganda, Madagascar, Zimbabwe, Mozambique, and Southern Sudan. And with regards to beliefs that there is low investment in research for treatments, so that to increase demand for vaccine, our partners had they assumed lack of investment in research for treatments. Some guys are saying, some users have been saying that this was done on purpose to increase the demand for a vaccine to boost profits. This was had in 3 countries: South Africa, Namibia, and Malawi. Seems to be a South African rumor. How did they manage to develop a vaccine in such a short time? Is it safe? This was a concern which was had in Kenya. And why are some vaccines labeled not for sale in USA, UK, and Australia? If they are not safe for them, why should Africa accept them? This was had in 3 countries: Kenya, South Africa, and Uganda. Next, please. With regards to effectiveness of the vaccine on the new variant, people are saying the new variant and the fact that public health recommendations are saying to keep wearing a mask and social distance even after you get a vaccine seems to undermine the perceived importance of the vaccine as a strategy to end the pandemic. This was had in South Africa, Kenya, and Zimbabwe. And finally, there was a debate which was generated by the former vice president of Zambia. Maybe some of you saw the video which went viral, where he said there was a need to test the vaccines locally to ensure the safety before rollout. So this some of the feedback that we are getting from our partner agencies who are working across the region. So it is this information that we use to come up with polls, which you will participate in the middle of this conversation. Thank you very much in advance. And This is what helps us come up with all these conversations for discussions. Thank you very much. Back to you, Wayne.
Thank you very much, Tole. Okay, now we're ready for our keynote speaker. As I said, Professor Bartholomew Akanmori of the World Health Organization. He's regional advisor for vaccine research and regulation, and he's based in Brazzaville, Congo. Dr. Akanmori will take us through the key points to keep in mind when you are looking at vaccine science and safety. Well, without further ado, Dr. Akanmori, welcome, and I toss over to you now.
Great, thank you. I'd like, first of all, to go straight to the point of just looking at development of COVID-19 vaccines, and to acknowledge that these vaccines have been developed pretty fast. It's taken a relatively short time, indeed shorter than usually it takes to develop them. However, to understand better how these vaccines were developed, to reassure the general public, in particular the media, about the development of these vaccines, I thought it was very important to put all of this into context. So in the next slide, I have a short plan. What I plan to do briefly is to talk about the major, to talk about clinical trials. In fact, and then to look at how specifically COVID-19 vaccines were developed, and then to go on and discuss safety and try and address other issues. But can we skip to the next slide, please? Right, so basically the focus will be on clinical trials. And it's important to say that clinical trials arise from a carefully designed plan, what we call a new drug development plan, a process which is carefully contrived of, planned, and a plan that in the next slide will show you the details. The draft development plan, as I said, yes, the drug development plan follows a carefully laid out pathway. It begins first with discovery of products. Then you progress to preclinical studies, or studies carried out without the involvement of human participants, and then on to the human development phase, or what we call clinical trials, and then which are, of course, also in phases, and then gradually moving towards product licensure, registration, and then there's a period after the product has been widely introduced to have what we call a post-approval safety monitoring of the product. Now, in this next slide, the development plan, the drug development plan, the elements of this plan, or what we call an investigational new drug application, are summarized. And I would like to take a bit of time to explain that this plan is developed well ahead of any vaccine testing. So this plan describes in detail what is intended, i.e. all the aspects of the product development, the entire process, including the target product profile, which means what will this product be used for? What effectiveness or efficacy is intended? What age category is this product meant for? All of these questions are answered in the investigational new drug plan. Now, in addition, it's important to point out that this is a prospective plan, i.e. it is developed long before a product even goes into testing and evaluation. And finally, the third aspect is that it's a logical sequence, stepwise process in which information around this product is accumulated, is evaluated, and may be changed over a period of time. Now, having said that, in the next slide, you see what the key elements of development of a product are. For example, a vaccine. The first important thing that a manufacturer establishes is activity. You have to make sure that what you are developing is a vaccine and nothing else, and that it does work like a vaccine. That's the first thing. And then the second thing is to ensure that, I think there's some background noise, is to ensure that the product is not toxic. It doesn't have any toxicity. Sorry, can you hear me?
Yes,
hello?
we can.
Yes,
Yeah,
Dr.
sorry.
Akanmori, we can hear you. There was just a participant who wasn't muted, but I think that person is muted. Sorry. Please continue.
Thank you. So then you have to make sure the product is safe and has no initial toxicity, because if there's any indication at that stage, you just stop the development because there's no need taking the product forward. Now, the third aspect is the quality of the product. You have to ensure that there's a process which meets international processes for manufacture of pharmaceuticals for that product. And there's a facility which can do this. And the final point is to make sure that the product is stable, stability over a duration of period, at least over the duration that the product has to be before its use. Now, the next slide now takes us to the, having understood clearly what goes into this plan. Now, in the next slide, in the next slide, I want to just briefly touch on the different phases that are earlier mentioned, i.e., I want to address the first phase where you look at, where you examine the basic constitution of the product to try and understand its the nature of the process, looking at that disease, looking at the disease itself, looking at the smallest level of a cell or a molecule, how this product behaves, and you try to establish all of this through basic research, again, without involvement of humans. Then comes the next stage where an investigation is carried out into the disease itself to better understand the disease. What will this product protect against? Will it protect against infection, i.e. the ability of the pathogen to get into the individual? Will it protect against disease, i.e. symptoms of the disease? What level of disease, severity, mild forms of the disease? Will it prevent hospitalization? And then finally, you try and look at the genetic composition and nature of the product, to understand better what this product is at the genetic level and to sequence it. And as you know, this is the first thing which was done with COVID-19 vaccines. And it's important to point out that most COVID-19 vaccines target a specific molecule of the pathogen, the disease-causing agent, the SARS-CoV-2 virus, namely the spike protein. So this is just to underscore why we go through these processes early on. In the next slide, please. So here, having described what happens at the very beginning, once a target has been identified and the research has been carried out, there is a proof of concept. The next thing is to characterize this product and to be able to manufacture it, and then obviously then to aim at testing it at different levels. It's important to know that in all of these, you want to compare the product you are developing against what is available for that disease. And in some cases, there may be nothing, as in the case of COVID-19 vaccines. And of course, you want to also make sure that new technologies that provide new ways of developing this product are applied to the development. And all of these can take months to years. But as we know, for COVID-19 vaccine, all of these were accomplished within a matter of months. Now, slide number 9. In slide number 9, quickly, just to show you a preclinical phase, what it is, what it means. These are studies in laboratory animals. Very often we insist on at least 2 different mammalian species, so you can have some comparisons. It also includes laboratory studies, and it includes looking at safety and looking at activity in these non-human species. And this slide shows you that it involves understanding characterizing the molecular or cellular nature of the product, and then also evaluating it in animals. And these are according to the guidelines of the International Council on Harmonization, or ICH. These are internationally acceptable guidelines that all manufacturers fulfill. Now, the next step is now moving into the what regulatory authorities do, for instance, for the FDA. Now, they would require that before you start developing a product and before you do clinical trials, that you require from them the permission for them to allow you to import and use a product in humans. i.e., you need to be exempted from importing a product which is not licensed and to evaluate it. That's the first thing. The second thing is that they request you need to also request for exemption from the regulatory authority to ship the product itself, even across countries from where it's made to another country. And then obviously it also requires that you create an ongoing file which is deposited with the regulatory authority, and you keep building it up with information that enables the regulators to carefully monitor the development process. This is critical because ultimately you're going to give the vaccine to healthy individuals who have no problems, no diseases, and just need protection. And you want to ensure that they do not receive something that would rather provide them with a health problem. Now, what are the key elements of this application? We talked about them. Proof of concept, i.e., is there activity? 2, you need to look at the target of this product and the indication. What is it going to be used for? You need to tell them what animal studies have been done? What did you find out? Is it safe to move to humans? You need to tell them also about the manufacturing. Is it manufactured under good manufacturing practice in a facility that qualifies to manufacture pharmaceuticals? And then you need to provide stability information. These are vital. And moving on to the next slide. So having done this, you then get to submit a full proposal. You make sure that you fulfill GMP. You make sure that all the guidelines are fulfilled, including the WHO guidelines, the ICH guidelines, good laboratory practices, the laboratories where every test associated with this product will be carried out. And I've just given you examples of these guidelines just to remind you that they exist. Now let's quickly go. Let's keep the slide number 13 and go quickly to 14, where we'll talk about clinical trials. Now, this is where I want to really focus on and let you understand that in carrying out clinical trials, there are distinct phases, phases 1, 2, and 3. They differ, one, in what they target. Phase 1 trials would target primarily safety in small numbers and with intense scrutiny over a very short duration, which means people are exposed to the product for a short period of time to estimate its safety. Then you move on to phase 2. Now, it's important to mention that for phase 1, there are about 20 to 50 people involved. One key point to mention is in clinical development of vaccines, you start with small studies with little knowledge about the product, and you progressively move to larger studies where you are accumulating more information about the product that would help establish its effectiveness and its safety while making sure that the quality is consistently the same. This is very important. And I think we need to understand how the complexities in product development and that no step can be sacrificed. Phase 2, you go to larger study. We are now trying to establish how effective it is. What is the efficacy? Can you increase the quantity of the product to find the best quantity to give you the best possible response? These are the questions you answer in phase 2. Then you go to the ultimate phase, phase 3, which is the last phase where you try to establish in a large trial involved thousands of people, tens of thousands, in some cases even up to 100,000, to establish how well the product works. This duration is usually the longest because you want to extend the period for as long as possible so you can better identify any safety issues. And secondly, you can establish a shelf life because ultimately this is the population that you believe will ultimately receive the product. You need to make sure that in designing this study, that you make sure that it is randomized and you do not know who receives what. There's no bias, and that's where randomization becomes critical. So we often refer to randomized controlled trials as the gold standard for clinical trials. And then, of course, the focus, as I said, is on effectiveness. And we're coming to the end. Now, let's go specifically to COVID-19 vaccines. Now, this figure here shows you exactly what happened with COVID-19 vaccines. Now, because of the time limit, the fact that these vaccines have to be developed quickly, it became important for the phases to overlap each other by compressing the timeline. So you begin with a phase 1, and then when you go into the phase 1 for a short period of time, you start a phase 2 at the same time so that they run concurrently, but you are monitoring. But when you're going to do this, you're going to speed up the trial, you need to put in place what we call risk-benefit assessments periodically, as many as possible at each stage. So in phase 1, you assess the benefits, you assess the risks, you look at the potential of occurrence, the benefits, the potential of occurrence of the risks, and you make a decision. This decision is made by regulators, and you do the same for phase 2. And this is exactly what happened with the development of these vaccines. So CEPI, the Coalition for Epidemic Preparedness Initiative, that's exactly what they did with the COVID-19 vaccines that are now being rolled out. They ensured that risk-benefit assessments were done by regulators, as I said, as often as possible, ensuring that there was no there was no compromise on the process. Now, let's try and answer some of the questions that were posed. One of the questions was COVID-19 vaccines. I want to summarize in saying they were carefully developed. One, all the steps in clinical development were fulfilled. Nothing was skipped. Two, that quality, safety, and efficacy were monitored throughout the studies. Four, that risk management and risk mitigation was a part of the process up to now. And so there are risk mitigation plans as these vaccines are being rolled out. In addition to just providing the vaccines. And then finally, now, where in Africa were clinical trials conducted for COVID-19 vaccines? So far, there are 3 countries that for sure carried out clinical trials. These are Kenya, South Africa. Indeed, for Egypt, they were trying to develop in collaboration with, in partnership with other developers. Their own trials are still ongoing. Then there was the other issue of what systems are in place in countries to ensure that COVID-19 vaccines, to ensure the safety of COVID-19 vaccines in Africa. Well, there are systems in countries. First of all, each country has a vaccine pharmacovigilance system, a system which was developed globally based on what we call the Global Vaccine Safety Blueprint. This blueprint outlines what countries should have in place, and we have ensured, WHO working with the countries, that they have these systems in place. And some of the elements I've spelled out. There's a national regulatory authority that will approve the vaccine and approve the risk management plan which accompanies each vaccine. Secondly, there's an immunization program, well-trained staff who will administer these vaccines. And they, in turn, will also require that if anybody has an adverse event or suffers any adverse event, that they are able to report it to them, that they collect this information. Of course, there are health facilities where people report these adverse events, and there are health workers who manage the events as well. So if you were to react because you are allergic to a product, you will be treated appropriately and you will recover invariably without any long-term disability. So these are all the stakeholders. Then there are disease surveillance officers in countries, disease surveillance and epidemiologists in countries who collect information around diseases, including vaccines and vaccine responses. And then finally, it's also important to stress that there are resources, and specifically for COVID-19 vaccine, WHO developed through the Global Advisory Committee on Vaccine Safety, a safety manual which is comprehensive, which outlines what the adverse events that countries should look for. It provides them with stepwise process of how to do this, how to transmit the data, how to look at severe adverse events, have them duly investigated, have a causality assessment, i.e. study them just as was done by the European Medicines Agency for the AstraZeneca Oxford vaccine, to make sure that there is no association or there's no causal relationship with the administration of the vaccine. This is vital. So these processes are in all countries. And I think I'll pause here so that we can have discussions. Thank you very much.
Thank you, Dr. Akanmori. Yes, there were questions, but on your tail end of your discussion, you answered some of the questions that were raised in the chat, like where we are on the road for an African vaccine. And we had also a question on why it's so fast, which you've answered. There's one more question, which I'll want to pose to you. But if you allow me, if we allow the next speaker to go, then you, after your presentation, you answer this, that would be fabulous. And the question is on, it's from Kate. She says, regarding the Oxford AstraZeneca vaccine and the 501Y-V2 variant, first discovered in South Africa, we know the study done here is limited in that it was small, only for people in their 30s. And even then, the results only apply to low efficacy in mild to moderate cases. What about cases, what about severe cases? Do we have studies and reliable data on that? So that is a question that will be coming back to you after Kate's presentation. Kate, to you.
Hi, everyone, and thank you for having me here. And it was really a pleasure to get to watch Dr. Akanmori's really informative presentation. So my job is to talk a little bit about the ways as journalists we can unpack the story of covering vaccines, specifically around the safety and efficacy data. So I won't take too much of your time, since I know many of you will have questions for Professor Akanmori, but thank you very much for getting my slides up, and we can begin with the next slide, please. Okay, so I'm just going to talk you through 7 main ways in which journalists can unpack the vaccine story effectively and quickly for your audiences. So number 1 is to get familiar with the safety and efficacy data, as I'm sure many of you already are. But the second part of this is to find ways to bring it home to your audience. And what do I mean by bring it home to your audience? Well, your audience will of course want to know what the data says, and they'll want to have it broken down and explained to them in easily digestible terms. So, and when I say bring it home, I mean make sure it's relevant to their context and setting. So for example, you know, when you're faced with this amount of data, sorry, could we go back a slide, please? No, back to number 1, thank you. When you're faced with this amount of data, which can be overwhelming, how do you unpack that and help it make sense for your audience? And so, you know, we have on this slide here, looking at the efficacy confidence intervals from major vaccine trials. And on the surface, when you look at the figures, you know, Moderna, Pfizer, biotech, which have been largely administered in the US and Europe, it does appear as if those vaccines have a higher efficacy rate than, for example, AstraZeneca, Oxford, Johnson & Johnson. But when you really unpack those figures, you see that the trials were conducted in different settings. AstraZeneca, Oxford, J&J was conducted in the presence of the B1351 variant. So the settings were slightly different. And from that, we can perhaps, you know, think about, well, if the conditions were exactly the same, would we be seeing that same high 94, 95% efficacy in the AstraZeneca J&J vaccines, we don't fully know how effective the Moderna Pfizer biotech vaccines are in the widespread presence of that variant, for example. So it's not an exact science in terms of comparing, and it's important to recognize the different contexts in which the vaccines were developed. And I think the bottom line is that if you look at the data on which vaccines prevented hospitalization, severe cases of COVID-19 and death, it's really 100% across the board. So all of the vaccines that were developed, nobody has died as a result of COVID-19 after they've received the vaccine, which is just fantastic news, and I think important to focus on when explaining to your audience. Another quick metaphor I wanted to bring in, it was really informative listening to Dr. Akanmori explain the process, and I hope that's been reassuring to many of you. There are metaphors that we can use when explaining this to our audiences. For example, a pretty common one that I've heard used is the traffic light metaphor. So the metaphor goes like this, that in a normal vaccine development process for something like malaria, for example, which has taken at least 30 years to get to the point now where we have vaccines, the process is pretty slow. There are lots of red lights. Imagine driving down a major highway, red lights, you're stopping all the time for protocols. In the midst of a global pandemic, many of those lights are green, so you get to go through faster. That doesn't mean you're not going through the same lights. It doesn't mean that the same protocols aren't there, but they are sped up because of the emergency situation. So I think that's quite an interesting metaphor for understanding why things can be faster and still be trustworthy. Next slide, please. Okay, tip number two is to follow the story of vaccine access, logistics, and equity. This is really important. Access to vaccines is about the interlinked elements of equity, democracy, governance and accountability, health as a human right, vaccine delivery, and logistics. So equity, as I'm sure many of you know, means that COVID-19 vaccines need to reach all groups of people, regardless of race, age, or other demographics, and they need to be affordable to poor and rich countries alike, not just those who can pay the most. The vaccine nationalism story has gotten a lot of play among journalists, quite rightly, and I think it's important to follow that. And to look at the ways in which COVID-19 vaccines are being delivered across sub-Saharan Africa. So the primary mechanism is the COVID-19 Vaccine Global Access Facility, also known as COVAX, which has at least 2 billion doses distributed in many countries in sub-Saharan Africa. The African Union is also distributing 1 billion doses, and then there are private donations and individual partnerships between countries and vaccine manufacturers. So it's just good to get a handle on exactly the mechanisms for distribution and which ones are happening in your country. I'll just jump to the next slide, please. Yeah, so keep on top of the rollout data. Here you see a map on the right, the source is BBC News, that is showing how quickly the vaccines are being distributed and where and which countries have yet to receive vaccines. So the countries with the darker color are those that have begun vaccinations. The lighter color are countries that have received vaccines. This was from a source about 2 days ago, so that's rapidly changing, of course. As you can see on the left, we have of some of the COVAX countries. That's not an exhaustive list. There are others, of course. And then there are other categories, so many countries have received AstraZeneca donations from India. Other countries have received donations of the Sinopharm. So, you know, it's interesting, I think, to keep on top of what is being rolled out and where and how quickly, and some of the challenges in doing so. Next slide, please. Yeah, so understanding vaccine confidence, this is a big one. And many of you may have come across people or stories who, you know, where people have said, I want to see more long-term data before I take the vaccine. I don't trust the vaccine. I've heard this rumor that says if you take the vaccine, it will be implanting a microchip in your body, blah, blah, blah. There are many kinds of rumors circulating. So, you know, we talk a lot as journalists about fact-checking rumors and correcting misinformation, but I also think it's really important to understand the reasons why people don't necessarily trust vaccines. And many people, of course, do, and actually the figures are looking much better than we had hoped, and are improving. However, when you do come across people in your stories who express some hesitancy about vaccines, it's good to understand the psychology of that and where it comes from. So the WHO describes vaccine hesitancy as a continuum, as you can see here in this nice diagram of an actual vaccine. So at the far left, we have people who typically refuse all vaccines, who don't trust them at all. On the far right, we have people who will accept all vaccines without asking any questions. And most people fall somewhere in the middle. Most people actually, I would say, are probably in the accept but unsure category to the right, where people will ask questions and want to know, quite rightly and understandably, about the safety, about the efficacy, about if the vaccines are safe for them and their families and their communities. But other people will be further down on the left. Well, they're, you know, refused but unsure, hesitant. And it's important to understand some of the reasons why, which I will explain in the next slide, please. Thank you. So there are different frameworks for understanding vaccine confidence. The Australian researcher Julie Leask, who sees vaccine confidence as a pyramid of acceptance made up of 5 levels. Again, as you can see, you know, the person who refuses all vaccines is actually pretty low, estimated less than 2% of people, and the unquestioning acceptor, estimated 30 to 40% of people. And so most people are kind of around this cautious acceptor level, the person who has some questions but trusts science and vaccine trial data, an estimated 25 to 35% of people. So according to the UK Royal Society and the British Academy, whose researchers study vaccine hesitancy, COVID-19 vaccine deployment faces an infodemic with misinformation often filling the knowledge void, characterized by either a distrust of science and selective use of expert authority. Number 2, distrust in pharmaceutical companies and government. Number 3, use of emotion. And number 4, echo chambers. And what does that mean? Well, basically that there are many factors that determine what level of trust communities have in vaccines. And those can include things like whether the health system has earned their trust in the past or not. You know, people who have not been treated in the ways they should have been by the health system in their country are much less likely to trust a vaccine. So these historical events impact people over time and often across generations. Other factors that impact vaccine hesitancy include whether people have access to the safety data and efficacy information, whether they've had access to positive human stories about the benefits of getting vaccinated, and so on. And as journalists, I think we're particularly well placed to help with the last 2. If people don't have access to the right information, if they don't hear about people who've had the vaccine and had positive, successful experiences, they are more likely to experience hesitancy. So as journalists, we have a lot of power in making sure that information is accessible to our audiences and serving our communities by sharing information that really empowers them to take care of their lives and their families and their communities. Slide number 6, please. Yeah, so, you know, I always like to come back to this. Remember the power of human stories. Numbers are compelling. For example, a story stating that over 300,000 people in Rwanda have so far received COVID-19 vaccines is wonderful. However, how many people will read past the headline? Human stories reach our hearts in a way that numbers don't necessarily. So here's a piece by UNHCR showing the first refugees receiving COVID-19 vaccinations in Rwanda. And this is at the Gashora Transit Center. So what does it mean to a refugee or to somebody in a high-risk group, somebody with comorbidities who has been, you know, staying at home much longer than another person might have been, somebody at high risk of severe illness from COVID, for example, for you know, people who necessarily didn't expect to receive a vaccine so quickly? How is that changing their daily lives? What does it mean? Does it mean they can go out to work? Does it mean they can go back and work in the market? And how do health workers feel after receiving a vaccine? You know, when interviewing them, some questions you can ask can go beyond the figures and ask, you know, what does it feel like to describe the sense of relief at being protected? So don't be afraid to explore the human spectrum of emotion while reporting on science. Science doesn't just have to mean data. Okay, and my last slide is, of course, you know, we always come back to this, continue to fact check and correct rumors, but acknowledge the reasons why people may believe in them. And here are 2 examples of a rumor on the left. You can test positive because of the vaccines. Of course, that is a myth. You cannot test positive because of the vaccines because there is no live virus in the vaccines. It cannot infect you. Another rumor on the right suggesting that viruses travel on 5G mobile networks, also debunked by WHO. So, you know, of course, fact checking is at the heart of our work as journalists. However, I would also suggest avoiding judgment or shaming people who believe in rumors, because those tactics often achieve the opposite reaction we're hoping for. So we can look to more constructive approaches like producing positive stories, like creating nuanced reporting. If we encounter vaccine hesitancy, interviewing an expert on that, for example, bringing in medical anthropologists to talk about the reasons why some communities may be hesitant, if you experience that in your reporting.
Kate, please, before you continue, there's a question from Sadia from Somalia. She says, a week ago, Somalia drove out the vaccination of people, the rollout of the first batch of Oxford AstraZeneca vaccines. Some people got the first dose, while health experts say the vaccine is not safe and causes blood clotting. So how does a journalist report this issue?
Thanks, Tole, for that great question. Yeah, so just to wrap up this slide, which is the last one. Yes, so just the point I was just wanting to make was that it's really important to produce a balanced story. So if, for example, you encounter folks who are more hesitant, it's great to make sure you also interview somebody from MOH or WHO or another organization working on the vaccines to make sure you also counter that with accurate information rather than risking sharing that too much in your reporting. So yes, and let me just come back to that question, Tole, one moment. Yeah, so I think this is a really great example of what I was just talking about, actually. So as Sadia says, you know, a week ago, Somalia drives out the vaccination. Some people received the first dose, while health experts said the vaccine is not safe and causes blood clotting. You know, this issue has got a lot of media attention. And I think it's important to, you know, when these things come up, which they will, make sure that your reporting is balanced, make sure that you talk to somebody who is able to actually give the data on the safety, right? So I think there have been some concerns in some countries about the blood clotting. However, I think that also there is data out there that suggests how few cases there have been within so many cases of people getting vaccinated. So putting this idea in context, putting the cases there have been of this in the wider context of how many doses have been delivered safely is really important. And I think just making sure you're not contributing to sensationalist reporting with your headlines, for example.
And if I may add, sure.
Yes, please, Dr. Akanmori, yes.
Yeah, Kate, I just want to quickly add that, as you recall, Sadia, when I went through the process of vaccine development, I mentioned that the role of regulators is to look at every aspect of a vaccine and right through from its development to its use. So if there's any issue, the regulators have the final say. They have the last say because they are the ones who are closest to the product. They received a plan for this product before it was developed. They have followed up. They have a file that contains every study which has been done with this product. And they continue to monitor this product in wider use. So they will definitely often carry out a causality assessment, look at the distribution of blood clots in populations where people on medication. In general, how many people do you have with blood clots in every million? And they will compare to those who received the vaccine and establish whether the blood clots were caused by the vaccine. And if the answer is no, it's no, and you have to trust the work of the regulators. And indeed, the Austrian Regulatory Authority has just announced, and Austria has resumed the use of this vaccine because the Austrian Regulatory Authority, the Medicines Agency, who approved these vaccines, also undertook these studies and ensured that there was no relationship between the jab and these blood clots. So we can just debunk this completely. And I think this should be what people should be looking out for, as Kate said. You listen to the experts, because anybody can make a claim about a vaccine not being safe. But are they the ones you should listen to? Perhaps not. It should be the regulators who approve the product. Over.
Yes, exactly. And there's actually a link in the chat that Anna has shared showing the study that actually shows that there is actually no increased blood clot risk with that vaccine. So that's a good link to click on. I think it's also good to look at, when something like this happens, as humans, we are quick to create this causal relationship and think if event A happens, it must be because of event B. And often that's not the case.
So before I send it to Wayne, there is a question for Dr. Akanmori. Journalists are asking about the effect of nutritional status of the vaccine in the body's ability to develop antibodies from the vaccine. Please clarify. And then before you answer that, Wayne had a number of questions. We are sorry, unfortunately, we are over time, but I know it's interesting. So for the folks who can continue joining us, please do. And the ones who will not, thank you very much. Wayne, to you.
Well, that is a good question for Dr. Akanmori. And I have a question. It sounds like a very simple question from Radio Wajir, community radio in Kenya. Simple question, but perhaps it's a good way to sort of put everything into focus. The question is, how safe is the COVID-19 vaccine?
Great questions. Very good questions. Yes, the first one about malnutrition and ability to respond to a vaccine? Clearly, vaccines typically, the average individual is able to respond to any vaccine, including people who may be malnourished. However, it's also important to point out that extreme malnutrition can also mean a less than optimal response in some cases. But nonetheless, the protection will be accorded even to people who are malnourished and even to people who may have underlying diseases that may affect the immune system, such as people living with HIV/AIDS. Invariably, they respond to vaccines and they are often also protected. Now, having said that, now quickly to the other point from Radio Wajir, I think it's about, if you can quickly remind me again, is how safe are the COVID-19 vaccines? They are safe. We know that for a fact, because we know that these vaccines have been rolled out into millions of people, and so far we have no cause to doubt their safety. There hasn't been any severe adverse event which has been investigated and found to be associated with the vaccines. Of course, there are side effects. There are mild side effects. People receive the jab, they feel pain, they get tired, they may have a fever, and they may, generally not feel, themselves, but invariably they recover uneventfully and without the need for any treatment. Therefore, these vaccines are safe and people should definitely receive their jabs when it's their turn, so we can make a difference, so we can have an impact on hospitalization, on severe disease, and ultimately, together with masks and other social distancing activities, we can reduce transmission and make life relatively easier for all of us. Over.
Thank you very much, Dr. Akanmori. I think we're running to the end of our time, and I would just very much like to thank Dr. Akanmori of WHO and Kate Thomas of Internews for joining us today. We'd love to hear from you journalists on this call about how you use this information that you got today, how you use it to produce stories. Please get in touch with us, share your stories with us, provide any feedback you like. You can write to the COVID-19 internews.org email that we included in your invitation to be in touch with us. I'll just let you know this dialogue has been recorded, so we will share that link with you so that you can go back and check your quotes to make sure they're all accurate. And I'll also let you know, maybe pencil this in your calendar on April 14th. We will have another media dialogue. This will be on vaccine disinformation, misinformation, and hesitancy with another expert from the WHO. And to end off, perhaps we could have Christophe show us the journalist poll that we've been working on. Christophe? There we go. So we would like the journalists on this call to let us know what they would like to see. This will be for a media dialogue that we will have after April 14th, because we already have our April 14th topic. But all of these ideas are good. We'd like to hear from you which one you would most like to see, and that's how we design our media panels. We make sure that we're fulfilling the needs of the journalist community in doing your daily work. So please submit your votes to this poll. And I think at this point, I'm going to thank everybody, say goodbye. Thanks again to Dr. Akanmori, to Kate Thomas, Tole, and we look forward to seeing you next month, April 14th. We will send an invitation to you all, and we look forward to getting your feedback on our panels. Thank you very much for being with us today.